Hi, my name is Jiyu, a third-year PhD student from the University of Melbourne, Australia. I am very glad to have this opportunity here to introduce our recent findings. We're working on a feasibility study towards exploration of automatic inconsistency detection for literature-based geontology annotation. Our objective is to develop an efficient and reliable automatic system for helping curators to detect inconsistent geontology annotation in biological databases. This is a simplified demographic of such kind of system. During application, the system will take a Go annotation record as input, extract its core component including experimental evidence sentence in the associated literature, the text attribute of geontology term, and the normalized gene symbol. Then these contents will be input into an automatic inconsistency detection or inference model. The model makes validation and flags any suspicious record to human curators for further review. The entire system is designed to satisfy a human-in-the-loop quality assurance setting. Here is an example of overspecific type inconsistency. It appears as the selected Go term overtells part of the gene function which is not supported by the literature evidence. If we refer to the geontology on the right-hand side, overspecific inconsistency often occurs as incorrect selection of a descendant on the ontological graph. As the example shows, the annotation using suckling behavior for gene TRH is incorrect because the evidence does not specify the process of food intake is absorption of nutrition from the breast. A better Go term shall be feeding behavior. We propose solutions to these three major challenges. The first challenge is, to the best of our knowledge, there is no systematic study on inconsistency characterization of Go annotation. Most of the papers were discussing the concerns of several database quality issues on a high level. Some papers do have a grounded studies, but we're focusing on sequence-based Go annotation. We reviewed and integrated several curation guidelines, orally communicate with expert database curators, and formalized four major types of literature-based inconsistency. These four inconsistencies can confuse either human or computer. The second challenge is there is no publicly available data with the annotation of typed literature-based inconsistency. It is infeasible to create such a dataset at this initial stage. To overcome this issue, we designed several heuristic rules to simulate type inconsistency in real world. The simulation is well accepted by expert curators. The last challenge is there is currently no existing approach or framework we can start or extend with. Thus, we propose a framework and top up with both rule-based and machine learning-based automatic approaches. We ensure the design of this framework is satisfying human-in-the-loop curation scenario and adaptive to the open-world knowledge. We evaluate the feasibility of the implementation through both empirical experiment and qualitative case studies. For the evaluation of the implementation, we focus on two aspects. One is the correctness, which evaluates whether the model can truly detect a typed inconsistency. The second is confidence, which evaluates whether the model is suitable for human-in-the-loop application setting. For example, if a model is less confident towards a prediction, then human can step in and make manual curation. The confidence is quantified using Shannon's entropy. The joint language model called GNN-BERT demonstrates the best performance in various aspects. It is good at detecting inconsistency regarding the Go specificity, good at capturing the subtle or small nuance of semantic differences, and also robust to out-of-distribution instances. The out-of-distribution instances denotes the species used for training and testing purpose are different. This perspective can evaluate whether an automatic approach is adaptive to open-world knowledge. This model can also maintain a decent correctness when low tolerance of uncertainty is set. We also conduct qualitative studies on real-world database. We apply the system to a database resource called Gene2Go provided by NCBI. A sample of identified inconsistency on the right is flagged as overspecific. It is probably because its experimental evidence does not give a concrete support to the gene function. To wrap up, in future, we're typically interested in enriching the characterization of Go annotation inconsistency. We will also focus on extending the feasibility of the system in real-world setting. We truly welcome curators to try our system and give us qualitative feedback. You may also find our early exploration paper here if you're interested in digging more details. This paper is much easier to start with. You're always welcome to chat with me during my poster session and send me an email. Thank you.